Hi, welcome to our first lecture on disease. Today we're going to talk about viruses and bacteria, and we're going to talk a little bit about their life cycle, and from that we're going to learn about how bacteria and viruses cause disease and methods of antibiotic resistance, and just learn a little bit more about the passage of disease through human populations. And this is going to tie into the second portion of the disease module, which will be later in the semester. So it's important for you to build the fundamentals now so that later on you have these fundamentals when we talk about disease further and when you do the SimBio module on disease. Okay, so do you remember our module on evolution? I attached the video where I discussed evolution in the biodiversity lecture, so go back to that if you need to review it. This is a summary of the observations that Darwin made and the inferences he drew from those observations that we consider now to be the basis of the requirements for evolution to occur. So, Darwin observed that individuals in a population exhibit a lot of variability in their traits. So we can see variability between us as humans, and of course this variability is present in all living organisms on Earth, ideally. And he also observed that organisms inherit traits from their parents. So because these traits are inheritable, they are selected against or selected for, depending on whether or not they are beneficial to that organism. So another observation he made is that populations produce more individuals than the environment can sustain. If there was an environment with plenty of resources and space for everybody, there would be no selection pressure against individuals. So there would be no selection for certain individuals to reproduce more effectively and to have their offspring live to reproduce again in a better way. So because of this environmental pressure, and because not all individuals can survive or reproduce effectively, there's going to be selection pressure, and the individuals who have traits in that variable soup of different traits, the individuals that have traits that are beneficial are going to be able to reproduce more effectively and live longer to reproduce more effectively, and therefore those individual traits are going to be selected for and will over time become more prevalent in that population. Oops. So humans have affected evolution of plants and animals themselves. So if we think about wild mustard, humans have evolved wild mustard to be all sorts of interesting plants so there are many examples of evolution around us, and we can see these examples in the plants and animals that surround us. So for example, all of the plants that you see here were evolved from wild mustard, so that was the common origin. So cabbage and Brussels sprouts and kale and all of those delicious things that we enjoy eating so very much. Now, similarly, humans have affected the evolution of dogs, and dogs have evolved from a common ancestor, but over time humans have bred dogs with traits that we found to be ideal or pleasant or desirable for some reason, so certain hunting traits or certain physical characteristics, and we have directed the evolution of dogs to have the physical characteristics or personality characteristics that we found most desirable. So here is a chart of that showing you the phylogeny of different dogs and how they are related to each other. So we are going to relate evolution to disease. Let's talk about a particular disease that is affecting humans. Sickle cell disease. So sickle cell disease is a disease where one part of a protein that's present in blood is mutated. So mutated means it was changed from some DNA nucleotide to another DNA nucleotide. And because of that very small change in that blood protein, when the blood cells are under pressure, they sickle. So you can see here on the image on the left, we have a sickled blood cell and a normal blood cell. So if you have sickle cell disease, it has a fairly poor outcome for the individual. So they have they struggle to bring oxygenated blood to all of their systems and organs, and this causes a lot of stress physically. So it's not an ideal condition to have. However, because of this particular mutation, individuals that have the sickle cell gene, or one copy of the gene that is mutated that causes these blood cells to sickle, is also protected against malaria infection. So this is known as the heterozygous advantage. So this happens when the heterozygotes, so heterozygotes have one copy of the diseased allele and one copy of a normal allele, and the allele is just the part of the DNA that encodes, in this case, the blood protein, but allele, we can just think about it as a gene. So some DNA sequence that is telling the cell to do something, in this case, make a protein. So when individuals that have one bad copy and one good copy actually have an advantage, that's known as the heterozygous advantage. And natural selection will, if we allow evolution to take its course, will select individuals that have one diseased allele and one non-diseased allele. So you might think to yourself, well, this probably is only the case where malaria exists. So if malaria doesn't exist, there is no advantage to having that one bad allele, because there is a high chance, there's a 25% chance that two individuals that have one bad copy of the gene each will have a child that has two bad copies of that disease, and therefore will have the full-blown effect of that disease. So let's look at where individuals distribute that have this heterozygous genetic makeup and compare that to the distribution of malaria-carrying parasites. So in the dotted areas here in gray, these are all the regions where the malaria-carrying vector are present. So we can see here, right in Africa and along some of the coastal regions here, malaria-carrying plasmodiums are present. Now let's look at the distribution of people that have one copy of that gene that causes sickle cell disease, and let's compare it to the distribution of the malaria presence. So we do see that in this region where malaria, in this very big region where malaria is present, we do see that more than 12% of the population have one of those diseased alleles. So we know that evolution has affected the distribution of these alleles and has selected for them. So people who had one bad allele have a reproductive and survival advantage. So mutations are just alterations of the DNA. This is just a little cartoon showing you that we can change the sequence of the DNA from one thing to another. Mutations can also be on a much larger scale than that, so you can lose part of your chromosome or you can lose whole genes. These kinds of things are a little bit more rare, but they do happen. So when you inherit your genetic material to your children, you are actually introducing some mutations and some changes in the chromosomes that your children, that will result in your children having a slightly different genetic composition than you do. So you're not inheriting your genes exactly 100% with fidelity. You are actually introducing some differences. So that's how genetic variability comes around. Okay, let's talk about genetic variability in viruses and bacteria and how viruses and bacteria evolve so rapidly to have genetic traits that allow them to adapt to an environment and have an advantage in an environment that changes or is adverse. For example, it's nice to think about these things on the spatial scale. So how big are we talking about when we talk about bacteria and viruses? So you might be 1.5 meters tall, maybe a little taller. A chicken egg is maybe 10 centimeters big. A frog egg is a few millimeters big. So here we've moved from meters to centimeters to millimeters. When we start talking about things in the micron scale, these are organisms that we need light microscopes usually to see, so they're about 10 microns or 1 micron, in that range. These will be bacteria. Now viruses will be much smaller than bacteria, and don't be confused by the cartoons that we show, because those are usually not shown to scale, but a virus might be somewhere down here in the maybe 80 or 90 nanometer range. So viruses are significantly smaller than bacteria. And in fact, it's an interesting philosophical question of whether or not viruses are alive, and you can ask yourself what does it mean to be alive once you learn about viruses, and decide whether you think viruses are living organisms or not. So here's a picture of a virus. This is a picture that was taken with an electron microscope. So because they are so small, we actually can't use conventional light microscopes to take pictures of viruses. We need to throw electrons onto viruses and see them that way. When the electrons bounce back, they create a picture that we can see the viruses with. And someone has pseudo-colored this picture, or basically painted it to have different colors. The electron microscope does not show different colors, so someone has artificially colored it. And here's a virus particle. We can see that it has some kind of envelope, and the inside is basically a few proteins, but mostly just nucleic acid. So these are very, very small, very, very simple things, right? Here are some pictures of different examples of viruses. They tend to have really nice organized exteriors, and it's a repeat of the same kind of protein. So if we look at this kind of virus here, that's one protein just repeated over and over and over again. So it makes this cylindrical shape. Here's again a different kind of protein repeated over and over again, and they assemble independently into these very beautiful structures. So viruses are kind of interesting to look at and beautiful. And they are obligate parasites, which means that they can't reproduce on their own. They require other cells, host cells, which they infect to reproduce. And how do viruses do this? So here's a picture of, again, probably an electron microscope picture that someone has pseudo-colored, so someone has artificially colored the picture to show you the different components of what you were looking at. And what we see here is a bacteriophage. So bacteria is bacteria, phage is virus, so it's a virus that infects bacteria. And we like, this picture comes up a lot because it's kind of a interesting alien shape or spaceship shape that people like to show because it's kind of visually appealing. So here's the bacteriophage, and we can see it on the outside of a bacteria cell, and it's injecting its DNA into the bacteria cell. So the bacteria cell is going to take up that DNA and unintentionally, or without maybe wanting to do so, it's going to propagate that DNA. So it's going to use all of its own machinery to make a lot of copies of that DNA. That DNA is going to tell the cell to make some proteins that are required to create more viruses. And then at some point, all of those viruses are going to come out of that bacteria cell and most likely kill it and spread a lot more viruses around. So let's look at a cartoon of that reproductive cycle. This is kind of a simplified idea, but this is how viruses work. So here's your virus on top, your simple organism here. It has capsid on the outside, that's that pretty repeating shell that it has, and then some nucleic acid on the inside. Sometimes it's DNA, not always, sometimes it's RNA. That gets into the cell. Now, DNA has all of the instructions of how to make an organism. So here's the viral DNA. All of the cell's machinery is actually propagating the DNA and making the proteins that the DNA instruct to make. So that's why we call them obligate parasites. So these are going to be reproduced inside the host cell. The host cell is making all of the components, and then at some point these proteins and nucleic acids come together again, and these viruses will either bud out of the cell and not kill it, or they might accumulate to such a level that the number of viruses inside the cell just take over and pop the cell and kill the cell on their way out. Let's talk about evolution of a particular kind of virus and how it has evolved to have drug resistance. So that's kind of what we're interested in for this module and the module that will subsequently follow. So here's HIV. We can see that much like the other cartoons that we've seen before, it's a very simple structure, so it has some outside. In this case, it also has those green knobs, which are proteins that have actually come off of the previous cell that they've infected, and these proteins are going to let that virus get into another cell to infect it. So it's kind of like a lock and key metaphor. So these proteins are going to be the lock to open up the cell that they infect to let them in. And inside we see a couple proteins and a little bit of nucleic acid. Again, very simple organisms. So much like the cartoon we saw before, we can go over the life cycle of this particular virus. So there's that lock and key in number 1, that those proteins on the outside of the virus are allowing that cell that it's infected to let them in. So that virus gets into the cell, deposits its DNA, and will take over the machinery of the cell. So now it's using the cell's machinery to replicate the DNA and create more proteins. And then at some point, the virus buds out of that cell again, and it takes part of that outside of the cell with it, so that next time it encounters another cell, that cell will let it in again because of those proteins. Those proteins on the outside are actually, in this case, human proteins. So another human cell is going to think this is not a infection, this is something that needs to get into the cell, and it's going to let it in. Now, people have come up with ways of combating HIV infection, and one of those ways was this drug 3TC. So we're not going to get into the entire life cycle of the HIV virus, but suffice it to say that there's a point where the genetic material of the virus needs to be copied into another language, so from RNA to DNA, and this drug blocks that. So the virus can still get into the cell, but its genetic language is never going to be translated so that it can make more viruses. So that's what the 3TC drug does. And what's kind of, scary and interesting at the same time is that the virus itself has evolved to be resistant to that drug. So what that means is at some point there was some mutation in the virus that meant that it would not, the drug would not block it anymore. So it's a mutation in this protein right here that the drug acts on. So this protein right here, actually, with that mutation that makes the virus drug resistant, with that mutation, the virus does not copy as well. So actually, it doesn't infect cells as well, that protein doesn't work as well, so the infections are going to be not as robust. That makes this virus that has this mutation weaker and less capable of reproducing well. So therefore, evolution will select against it. It's not going to reproduce as well, so it'll be less common. But once you introduce that drug, all of a sudden, this type of virus that has a mutation in that protein has an advantage. And over time, we are going to kill off all of the viruses that don't have the mutation that make it resistant, and over time, the virus that has the resistant mutation is going to propagate and take over. So in several patients, people have looked at the percent of HIV that has this mutation that makes it resistant versus the others. And we can see it starts off very low because, like I said, that mutation actually is not necessarily good for the virus on a regular basis. But in the presence of that drug, all of a sudden, that mutation is present in all of the viruses because they are all originated from the original viruses that were originally present in a very small amount. So that's how drug resistance works. Over time, a small part of the population that is resistant is going to take over and become the dominant virus or bacteria. I just want to take this opportunity to remind you that smallpox is a virus, and you will have in the future some reading about smallpox, and we're going to learn a lot about vaccines and smallpox and the history of smallpox and how it is a disease that we've essentially eradicated. And these are more electron microscope pictures. So now they're taking cells and they're kind of slicing them in half with a knife so we can see kind of a cutaway of the cell, the inside of the cell. And they're showing you that cells that are infected with smallpox create these granules that contain the virus, these inclusion bodies that are very characteristic of cells that are infected with smallpox if you look at them under a microscope. So of course viruses have a significant effect on the health of our agriculture and our animals, as well as our own effect, as well as an effect on our own health. So we are very interested in learning more about viruses and their life cycle and what makes them resistant to various viruses are not subject to antibiotics, but resistant to various treatments. And of course, one virus that affects humans very commonly is the cold, or is the flu. And the flu can have a range of degrees of difficulty, varying degrees in severity. And in 1918, there was a flu pandemic that killed over 1 million people, and this was a massive health catastrophe in the United States and in other parts of the world. And we're going to talk a little bit more about vaccines and how vaccines work and how vaccines work in the context of viruses. But the point of this is just to say these things are affecting us, and we need to keep in mind all of these different elements. So drug resistance, viral life strategies, and vaccines, and all of these elements that we consider important to our health. All right, see you in the next part. Thank you.